Hi everyone. In this video, we're going to look at the components of the endomembrane system and how they function together in eukaryotic cells. The term endo refers to within. So when we talk about the endomembrane system, we're talking about a network of membrane-bound structures that function together inside a cell. The endomembrane system has multiple functions, including the production and modification of proteins, synthesis and storage of cellular components, and the breakdown of pathogens or old and worn-out organelles. These are the major components of the endomembrane system, as shown on an animal cell model, along with a brief synopsis of their functions. Through the rest of this video, we're going to zoom into each one in turn to get a better idea of the role that they play in an animal cell. We're going to start our journey through the endomembrane system here with the nuclear membrane. As you can see from the model here, it's a double membrane, which means two phospholipid bilayers, as we looked at in the previous section on macromolecules. Throughout the double membrane, you can see these pores, essentially holes that allow controlled transport of materials like RNA into and out of the nucleus. You can also see that the nuclear membrane is continuous with the rough endoplasmic reticulum, which we'll move on to next. The endoplasmic reticulum, or ER, exists in two forms, each with its own functions. We'll start with the rough ER, which is continuous with the nuclear membrane, as we previously mentioned. It's called rough because of the ribosomes on the surface, shown here as brown spheres. As with the cytosolic ribosomes, the job of the bound ribosomes is to synthesize proteins from instructions in messenger RNA. The difference between these ribosomes and those in the cytosol is the destination of those proteins. The proteins produced in the rough ER are those which will either be embedded into a membrane or will be secreted from the cell. The smooth ER, on the other hand, has a tube-like appearance and has no ribosomes. The smooth ER is like the liver of the cell, and as such is critical for detoxification and the production of lipids. It also plays an important role in cell signaling due to its role as a calcium ion store, and we'll discuss this more in the section on cell communication. Our next structure is the Golgi apparatus, which is shown on the model here as this lilac-colored stack of membranes. The Golgi receives proteins from the ER via transport vesicles, like this one, circled in purple. Vesicles also allow materials to be passed from one stack of membrane to the next, starting at the cis face and progressing to the trans face of the structure, receiving any required modifications in the process. From here, more vesicles transport those modified proteins to their correct cellular location. In this way, you can equate the Golgi with FedEx for the cell, receiving packages and sending them to their various destinations. The plasma membrane is one of the components that are found in every single cell. In every cell, the plasma membrane, like the membranes comprising the other structures in a eukaryotic cell, is composed mostly of phospholipids, like this one shown here. The concurrent hydrophilic and hydrophobic nature of these molecules means that they automatically form a bilayer in aqueous environments like cells. If we zoom into the membrane, we can see multiple proteins embedded in the membrane. And the exact protein composition will help to determine many of the specific functions that can be accomplished, like transporting molecules into and out of the cell, as well as relaying signals from the environment. Let's have a look at the direction of transport of molecules through these components of the endomembrane system. We'll start here at the rough ER, where proteins, such as insulin, for example, will be made and packaged into a vesicle for transport to the Golgi. Once the vesicle arrives at the cis face, the proteins will be passed through the stacks by other vesicles, receiving any modifications as it goes, until it reaches the trans face and is packaged into another vesicle. This vesicle will be transported to the plasma membrane, where it will fuse with the membrane and release its contents out of the cell. Lysosomes are typically only found in animal cells and are not usually present in plant cells. The lysosome is another component of the endomembrane system, but it doesn't function in the production or transport of proteins. Instead, It serves as a digestive system for a cell. Let's consider a situation like a bacterial infection inside your body. You have specialized types of white blood cells whose job it is to get rid of those bacteria, and they start to do that with a process called phagocytosis, kind of like cellular eating. They use the plasma membrane to envelop the bacterium until the membrane pinches off into a vesicle. That vesicle will be transported to and fused with a lysosome, a membrane sac containing acid and hydrolytic enzymes. Once inside the lysosome, the bacteria will be broken down by the enzymes, and waste products can then be secreted out of the cell. A similar process called autophagy or self-eating is used for cells to break down old or worn-out proteins, cellular components, and even organelles, so that the molecules can then be recycled into new components. The final component of the endomembrane system is vacuoles, and this is another major difference between plant and animal cells. In animal cells, as shown at the top here, the vacuoles are small and tend to come and go rather than being a permanent fixture in the cell. They're used for short-term storage of nutrients, water, or waste products. In plant cells, as shown at the bottom here, there is a much larger permanent vacuole called the central vacuole. It's often the most prominent organelle in a plant cell, and this is usually what we're referring to when we talk about vacuoles in cells. It has a really important role in maintaining water balance in the cell. We'll look at much more in a later section. It's also necessary for water storage, along with other molecules like nutrients, pigments, and even toxins, to keep them away from the rest of the cell. Thanks for watching, and please make sure to check out the other videos in this section.